I'm Dr. Amit Bhattacharya, Associate Professor at Department of Zoology, Ramjas College, University of Delhi. And today I will be talking about a topic which is chromosome analysis and structural aberrations. So in the part of this lecture, we will be talking about what exactly the chromosomes made up of, and why do we require the chromosomal analysis, and how can we decipher or find out what are the structural aberration or the structural changes in the chromosomes occurred. So before starting this talk, we will talk about the basics of the chromosome, what exactly the chromosomes are made up of. We will understand its dynamics, and then we will move on to the structural aberrations or the changes which comes under the chromosomes. Now, as we know, the chromosomes plays a very integral part of each of the cells. So they are present within a specialized organelle called as the nucleus. Now these chromosomes are made up of DNA, which is deoxyribonucleic acid. Now the deoxyribonucleic acid is basically a molecule that carries the genetic information for the development and functional of an organism. It is made up of two strands which are interlinked to one another with the help of the bonds which are there. So many a times it is referred as the double helix structure. Each of these strands are made up of sugar backbone along with a phosphate group. So the sugar in this particular DNA are basically the deoxyribose sugar. They contain four bases, main bases, which is adenine, cytosine, guanine or thymine, which is present. So they are referred in the short form as A, C, G and T over here. Now the DNA packaging into the nucleosome and the chromatin is very important for the understanding. Now the DNA which is present as the double helix structure, it subsequently coils up and it forms into a compact structure which is called as the chromosome over there. So we can see over here in the DNA packaging, the nucleosomes are there and subsequently the chromosomes are formed in a package form. We will come into the understanding of them in the subsequent slides. Now the chromosomal DNA is basically a double helix structure in which we can see there are two helix which are running anti-parallel to each other. Each of them are attached to one another with the help of the hydrogen bonds between the nucleotides which is present. So the nucleotides can be adenine, thymine, guanine and cytosine which are present. So these nucleotides have got the characteristic hydrogen bond formations between them. So we see over here some of the nucleotides and how the hydrogen bond formation are taking place. So we can see the adenine base pairs with the thymine over here, and the guanine base pair with the cytosine over here. So A pairs with T and the G pairs with C. So this is called as the complementary base pairing which occurs in the DNA double helix structure. So that's how the DNA double helix is formed. Now this DNA double helix is very important. Now it is said that a haploid human genome contains approximately 3 billion base pairs of DNA which is packaged in 23 chromosomes in the case of haploid human genome. Now there are total about 6 billion base pairs of DNA per cells which are present, and it is said that the human body contains about 50 trillion cells. So if we work out, it comes out that about 100 trillion meters of DNA per human is present. So you can imagine how large content of the DNA is present within us, but over a period of time they have been characterized and condensed into specialized structures. That's why they form such microscopic structures. Now if we see this diagram over here, this diagram shows how the DNA double helix structure have been condensed first onto the histones, then subsequently supercoiled and then formed into the chromosomes over here. Now the chromosomal DNA packaging is occurring within the microscopic nuclei. So what happens is, in the first level of the packaging, they are packaged onto specialized protein molecules which are called as the histones. Now the histones are basically positively charged proteins, so that's why they strongly adhere to the negatively charged DNA and form the complex which is called as the nucleosomes over there. Now these nucleosomes are the specialized structure in which the DNA is coiled onto the histone octamer over there. Now, the nucleosome fold up to form a 30 nanometer chromatin fiber which forms loop averaging about 300 nanometer in length. The 300 nanometer fibers are compressed further to fold and form a 250 nanometer wide fiber which is tightly coiled into the chromatin of a chromosome. So, we can see over here how exactly the DNA is coiled onto the histone molecule, which is a octamer molecule, and how it is being further packaged to form the DNA, the chromosomal structures. Now, the histones are basically a family of small positive charged proteins which are termed as H1, H2A, H2B, H3 and H4. Now, as the DNA is negatively charged due to the presence of the phosphate groups in its phosphate sugar backbone, so the histone bind tightly with the DNA structures because the histones are positively charged while the DNA is a negatively charged molecule. Now, it is being said that in plants and animals, including the humans, the chromosome reside inside the nucleus of the cell, and humans have about 22 pairs, numbered pairs of chromosomes which are called as the autosomes, and one set or one pair of chromosome which is called as the sex chromosome, which can be XX or XY. So, the total comes up to 46 chromosomes in the case of the humans. Each pair of the pair contains two chromosomes, one coming from each of the parents. So, which means that the child inherit half of their chromosomes from their mother and the other half from the father in that particular case. Now, the chromosomes in the case of the bacteria are about one or two circular chromosomes which are present. Now, each species of the plants and the animals have got a different set of chromosomes and the numbers varies up. So, we will take up some of the examples how the numbers varies up. So, if we see the rice plant, it has got about 12 pairs of chromosomes, while in the case of the fruit fly or the Drosophila, there are 4 pairs of chromosomes. In the case of the dog, there are 39 chromosomes which are there. So, in the humans, there are 23 pairs of chromosome which includes the sex chromosome, while the first pairs are called 22 pairs are called as the autosomes over here. Now, the autosomes are the chromosomes which are present, which are 22 numbered pairs of chromosome that most of us carry in almost all parts of the cell in our body. The largest of the autosome is basically the chromosome 1, which contains approximately about 2800 genes, while the smallest autosomes, which is the chromosome 22, contains approximately 750 genes in our case. So, if we see the diagram over here, this is a diagram which is showing all the 22 pairs of the autosomes over here, and at the end, if we can see, there is a set of sex chromosome which can be XY or XX over here. So, this is the set of the sex chromosomes which is present. Now, the X chromosomes, if we comes, the X chromosomes are basically the last set of the chromosome which is present, which are mainly comes under the sex chromosomes, while the rest of the 22 pairs are called as the autosomes. So, the biological female individuals have got 2 X chromosomes. So, many a times they are referred as XX, while in the case of the male there is 1 X chromosome and 1 Y chromosome, while there are some exceptions are also there to this particular rule. Now, the female inherit the 1 X chromosome from their father, while the other X chromosomes comes from the mother over there. While in the most of the cases in the biological, male people always inherit their X chromosomes from their mother. Now, the X chromosome is about 3 times larger than the Y chromosomes and contains about 900 genes, while in the case of the Y chromosomes, which is present in the males, it has about 55 genes present in them. Now, the Y chromosomes are also a part of the sex chromosomes in the cells. Now, the females, as we just discussed, have got 2 X chromosomes, while the males have got 1 X and 1 Y chromosome. The Y chromosome is a very small chromosome and it likely contains about 50 to 60 genes which is present in this particular thing. Now, one of the important part within these chromosomes is called as the centromeres. Now, the centromeres are a point within the chromosome where 2 of the chromatid arms connect to one another. okay, or they get joined over there. Now, the centromere was first described by the German biologist Walter Fleming in the year 1880s. Initially, it was called as the primary constriction of the chromosome. Now, the centromeres are such an important part of the chromosome because if they are not present, the cells are unable to divide properly, because in the centromeres only the spindle fiber comes and attaches to them, then only the process of mitosis or the meiosis initiates or takes place. And if it is not present, without the centromere, the cells cannot divide properly and the overall process of the division fails in those particular cells. Now, on the basis of the centromeres, the chromosomes have been classified into 4 major types. The first one is called as the metacentric, next comes the submetacentric, the third one is called as the acrocentric, and the fourth one is called as the telocentric chromosomes. Now, the centromere position is very important and it forms the important part of the attachment of the spindle fiber for the division process. Now, we see the diagram over here. This shows the positioning of the centromere. So, we can see the right hand side image, which is a metacentric chromosome where both the arms, with the P arm and the Q arm, are almost same length. So, that is metacentric. Submetacentric, it slightly moves to one of the side and one of the arm is slightly shorter than the other arm. Then comes the acrocentric, it is almost present at the end, and when it is present at the end, they are called as the telocentric chromosome because they are present at the end position or the telomeric position over there. Now, the acrocentric chromosome, one of the arm is extremely small while the other is extremely long, while in the case of the telocentric chromosome, one arm is missing because the centromere has moved to the end of one of the chromosomal part over there. Now, this is the karyotype of a normal female present over here. So, the 46 chromosomes are present if we see, and there are 2 X chromosomes which are there. So, in total there are 46 chromosomes which are there, in which the first 22 pairs are called as the autosome and the last pair is called as the sex chromosome over there. While if we see the example or the picture of the normal male karyotype over here, it also contains the 46 chromosomes over here and the XY is present. So, we can see all the chromosomes are present over here, the autosomes, and at the end there are XY chromosome, which is basically the sex chromosomes which is present over here. Now, with the help of understanding these particular chromosomes, a study came up which was called as the cytogenetics. So, cyto means coloration and genetics means understanding the genetic information and then the main sources of these genetic information present within the bodies, genes, chromosomes and these things. Now, traditionally the term cytogenetics was merely used to describe the investigation of the chromosomal abnormalities and how they are linked to certain diseases. So, we can see over here the image which is shown over here, it shows that how the chromosomes of a single cell are scattered in that particular plate, and by observing them we can make out that which part of these chromosomes are missing, and then we can trace out which disease this particular person or the child is suffering from. So, the cytogenetics plays a very important role in staining these chromosomes and subsequently reading them or translating them into proper information that what kind of chromosomal abnormalities, which can be in the number of the chromosome or the structural abnormalities in the chromosome which can occur, and then we can link to a certain disease condition. Now, one of the initial examples of the cytogenetics came up with the discovery of the Down syndrome. So, in this particular disease, there were numerous diseases apart from the Down syndrome which has been linked to the chromosomal abnormalities. So, the Down syndrome, we will take up the example of the Down syndrome, was discovered in the 1960s and how the chromosomal defect was causing a developmental problem in that particular patient who was having the Down syndrome. Now, the chromosomal analysis plays a very important role nowadays. Now, the chromosomal analysis basically means that the scientist or the lab technicians, they look for the chromosomes in the sample of the cells which they have collected from the person or the patient, and then they start identifying the genetic abnormality which can be caused in that particular sample. So, the number of the chromosomes are counted, and then the structures of those chromosomes are checked or mapped along with the standard maps which are present. And if there is any abnormality, it can easily decipher out that this particular disease condition the sample is having. Now, during the pregnancy, the cells from the amniotic fluid are also used for the testings. Okay. So, this is collected from a chorionic villus sample which is called as the CVS or the amniocentesis. Now, this process helps to check that whether the newborn baby which will be delivered soon will be having some kind of genetic or the chromosomal abnormalities or not. So, this is one of the oldest methods for obtaining the DNA of an unborn child. So, the amniocentesis, what it is done is the samples are collected with the help of the needle over there. So, the fluid is collected, and then that fluid is treated with the various chemical treatments, and subsequently they are lodged onto the slide where these cells gets ruptured out due to the chemical treatments and the hypotonic treatment over there, and these chromosomes are then observed under the microscope after proper staining, and then a karyotype is prepared to decipher out what are the chromosomal analysis or the prenatal screening which is being done. So, this prenatal screening plays a very important role during the pregnancy, that to check before the delivery that whether the unborn child has got some kind of chromosomal aberrations or some kind of chromosomal defects which is there or not. Now, the cytogenetic analysis protocols involves various steps which are shown over here. So, we will come into this one, how exactly these steps helps to decipher out the chromosomal analysis. Now, in this particular thing, what happens is, first the blood samples are collected from the patients, then it is cultured with the help of the growth stimulations for 3 days at 37 degrees Celsius. Then a chemical which is called as the colchicine is added at the end of the culture period, which helps in the arresting of the cell in the metaphase. Now, the metaphase phase is that part of the phase of the mitosis where the cells chromosomes are best visible, because the chromosomes are properly condensed and they are easily visible within the microscope, through the microscope. Now, once the colchicine is added, what happens is there is a cell arrest taking place in the metaphase. Now, there is hypotonic treatments which are given, the cells are dropped onto the slides and the metaphase plates are being prepared. They are being stained and observed under the microscope or the machines through which the images are taken. Then subsequently the karyotyping is being done, or a FISH method is done, which is called as the fluorescent in situ hybridization method. Now, in this particular karyotyping, what happens is the cytogenomic tool is being used to examine the chromosomal material under a microscope. So, we just discussed what happens is the samples are taken in, then there are centrifugations are being done, various chemical treatment, hypotonic treatments are being done, and then it is lodged onto the slide, and subsequently they are observed under the microscope to see the diverse cells over there. Now, these cells are then plated and the images are taken up, which are then subsequently then they are dispersed to form the karyotype or the images of the chromosome, which are then compared with the standards chromosome images which are present. And what happens is, during the staining process, each of the chromosome gives a definite patterns which is there. So, there are bands and interbands which are there, which are then compared with the standard bands and interbands of the chromosomes, and then it can be deciphered out whether the person is suffering from some kind of chromosomal aberration or there is some kind of chromosomal number change which has occurred in that particular sample or not. Now, this particular karyotyping, it helps in understanding the basic information about what kind of changes which has occurred in that particular person. Now, these helps to count the number of the chromosomes also. Now, as we count the number of chromosomes, if there is any change in the chromosome number, it can be easily deciphered out. Like suppose when we count the chromosome number in the karyotype, if there is any missing chromosome or deleted part of the chromosome, it can be easily deciphered out, and then the person can be said that this particular kind of abnormality has occurred in that particular cases. Now, in the next part of the lecture, we will be discussing about the different types of the chromosomal aberrations which are there, which includes the changes in the chromosomal number. We will see some of the patterns where there are trisomy which is there, means the trisomy 18, trisomy 21, which means that instead of normal 2 chromosomes or a pair of chromosome, the patient or the person is having 3 chromosomes. And we will discuss about why exactly these particular chromosomal abnormalities or the change in the chromosome numbers are occurring in these particular patient, and how these karyotypings images are helping to decipher out or find out or locate the exact location where these particular things are occurring. But this karyotyping has now become a very integral part of the latest researches also, because in most of the cancer cases we know that there are chromosomal aberrations which are taking place. For example, in the case of the leukemia. Now, in the leukemia, it has been found that in many of the cases there are chromosomal translocation or chromosomal part deletion which takes place, which means that a chromosome part from one of the chromosome translocates or moves from one chromosome to the other chromosome. So, this is called as the translocation part. Or there are deletions which are there, which means that one of the chromosomal part has been deleted. So, this karyotyping or the chromosomal analysis plays a very important role now in understanding the diseases also which are coming up. So, with this note, I come to the end of the first part of the lecture. In the next part of the lecture, we will be discussing more about these current approaches to genetic testing and the karyotyping. Thank you very much.